Hello, anatomy students. Now that you know about the anatomy of the heart and the path of blood as it flows through the heart, let's talk a little bit about how the heart is able to pump that blood each and every day through your body. So, we're going to talk about cardiac muscle and the cardiac conduction system. So, cardiomyocytes is the term that is used to name the cells that make up the myocardium of the heart. And they have a very unique structure. First of all, they're always performing aerobic respiration because they need to generate lots of ATP in order to contract as often as they do. These cells also are connected to each other through a specialized structure called an intercalated disc. And the intercalated disc is described as an interdigitating fold, which basically means that the intercalated disc on one cell has these folds that look like these fingers on my hand, and then the adjacent cell is going to interlock with them like so, just how the fingers of your hands can do. But these cells also have other connectors or junctions. They have mechanical junctions, which are proteins called desmosomes. At least there's multiple proteins that are going to make up a desmosome, but you can think of it as these proteins that just kind of connect the two cells one to another, and they can hold on to each other to keep them from separating while they're doing all this contracting all the time. And then we also have electrical junctions, which are called gap junctions. The gap junctions are these tiny little tunnels that are going to allow ions to flow from one cell to the next without interruption, so that you have swift, efficient communication, and all of these cardiomyocytes are going to communicate with each other so well. They're almost acting as if they are one cell, as if only one cell makes up the heart. So that's the importance of these gap junctions. So when we talk about how the heart is able to contract, you're going to be hearing terms like depolarization and repolarization, very similar to how neurons depolarize and repolarize. The heart is going to do the same thing. All of this electrical activity is going to start in a specialized area of the right atrium, which is called the SA node, which is right by the number 1 there. That's called the pacemaker of the heart because this is the area that's going to generate all of the electrical activity that allows for a heartbeat to occur. The cells that make up the SA node are kind of part cardiomyocyte, part nerve cells. So it's a very highly specialized area. And we're going to talk about why the SA node is able to constantly create this stimulus each and every day, one right after another, without rest. So here we go. This is how the electrical signal travels through the heart. Number 1, our SA node is going to generate the signal, and it travels through the right atrium and across the interatrial septum, which is right over here. It's going to flow into the left atrium. Now, once these cells depolarize, the atria will contract, but what I wanted to make mention is that the left atrium is going to contract a little bit after the right atrium because that signal has to travel a little bit further in order to reach the left atrium. Once the atria depolarize, the signal reaches the AV node, which is here at number 3. Again, it's atrioventricular, so it's in between the right atrium and the right ventricle. And then it's going to send its impulse over to where the number 4 is. This is called the AV bundle or the atrioventricular bundle. And you'll see that the AV bundle forks off into 2 branches. These branches travel down the interventricular septum, and then they whip around the apex of the heart and around the walls of our ventricles. These are called the right and left bundle branches, and the parts that actually creep up the sides of the ventricles are called the Purkinje fibers. And then the Purkinje fibers will distribute excitation through the ventricular myocardium. You may be interested to know that both the sympathetic division and the parasympathetic division of our nervous system innervate the heart. And so, our brains can affect the speed or the rate at which our heart is contracting. So, if you are under stress or if you feel threatened, even if you feel threatened because you have to create a flip grid for anatomy class, then, that could start your heart pumping maybe a little bit faster than what it normally would. So that would be your sympathetic division. Your parasympathetic division is going to do the opposite, and it will slow your heart rate down. Okay, let's talk about the electrical and contractile activity of the heart. All right, I told you before that I was going to show you why this sinoatrial node is able to continuously generate a signal, and that's because it does not have a stable resting potential. So resting potential for a neuron, you may recall, is negative 70 millivolts, and the sodium potassium pump was always working after hyperpolarization to try and get that cell back up to its resting potential. There's none of that happening in the SA node, and as you can see, we have slow sodium inflow, so sodium is coming into the cell, but then all of a sudden we have calcium and sodium inflow. So because we've got two positive ions coming into the cells in the SA node area, we have a pretty sharp positive slope here showing us that our membrane potential is going up. And then on the downside, our potassium ions are going to go out. And because we have some more positive ions leaving the cell, that's going to make our membrane potential become more negative. And then we have a slow sodium inflow again, and then the whole thing just starts all over again. Notice that our action potential is happening here once we have calcium and sodium in the cells, and then our potassium is flowing out. And so that's where the action potential is happening. So each time that we get depolarization of our SA node, each time we actually have our calcium going in and our sodium going in, when we have that depolarization happening, that is one heartbeat. So this is happening all the time. All right, so the reason why our SA node has a different action potential is because the ions are going to flow from cell to cell through the gap junctions. And that's also why the SA node is able to propagate or send out these signals so darn quickly. The amount of gap junctions that you have in between the cardiomyocytes is what determines the rate at which the propagation of the signal occurs. So for instance, the fastest propagation is down by our Purkinje fibers, so along the walls of our ventricles. And the reason for that is because those cardiomyocytes have many, many more gap junctions to allow for an easier, faster flow of ions from one cell to the next. And so that signal gets communicated way faster than the AV node up here, which again is found between the right atrium and the right ventricle. And it's not that it's bad or good, it's just that it just is. The fact that we have slower propagation here by the AV node means that our ventricles are going to have time to fill completely before they contract and send blood to the body or to the lungs, again, depending on which ventricle it is that you're actually talking about. Okay, so this is just showing us a little bit about how our cardiomyocytes are going to contract. So because calcium ions, which are positively charged, play a role here, this is going to allow these cells to contract for a longer period of time than perhaps they would be able to normally. And so let's just take a look at the yellow number 1 that I have in the picture here. First, our voltage-gated sodium channels are going to open, and that is going to cause a rapid depolarization. So you can see how rapid it is. This line is completely vertical. There's not even a slope to it. This is going to give us myocardial contractions, so that's what's happening here with our red line. This is showing us our myocardial contraction. I also just wanted to make mention that we also have an absolute refractory period here, which basically means that while this is going on, we're not going to be able to get like another signal coming through unless there's something wrong with the conduction system, which we'll talk about in the future. All right, all the way up at number 3 is where our sodium channels are going to snap shut. But what happens at number 4 is something a little bit different. We have our calcium channels that are going to open, and calcium is going to start flowing in while potassium is going to start flowing out. So again, we've got this plateau here because we have an exchange of two positively charged ions. You see, if we didn't have calcium coming in here, we would have a very sharp repolarization as the sodium was, or as the the potassium was flowing out of the cell. But we don't. We have this plateau here, and this is what is going to elongate the time that our cardiomyocyte is contracted. At about number 5, we have our calcium ion channels closing. Calcium is now going to be pumped out of the cell, which is going to make this repolarization happen a lot faster. So we have a much steeper negative slope here on this side of our curve. And again, here's our absolute refractory period. So we're not going to be able to kind of stop or start another heartbeat while this blue, like in the time frame of this blue box. Again, unless there's something wrong with the conduction system. All right, so this might be a shape that you are familiar with. This is an ECG or an EKG. I just wanted to make mention, though, that this single trace that you see in this picture here, this is kind of like an average of all of the electrical activity that's happening in the heart. Like this is something that you would get from, when I was on the ambulance, we used to use these old 3 leads where we would just put a couple of leads on the patient's chest. But, what they do now are 12 leads in order to record the electrical activity of the heart, like in all places of the heart. And so now you have to look at like almost 12 different tracings almost in order to get a really good picture of what's going on with the heart. But we're just going to start with like the introductory ECG. ECGs are also called EKGs. So those two terms are synonymous. So systole and diastole are going to refer to contraction and relaxation of the heart, respectively. Normally, when clinicians use systole and diastole, they're talking about ventricular contraction and ventricular relaxation, but it could refer to atrial contraction and relaxation as well. But then it's usually termed atrial systole and atrial diastole. So any deviation from this pattern is something that's called an arrhythmia, and it's typically caused by an interruption in the electrical conduction system of the heart. So let me move myself out of the way. All right, so atrial activity on this ECG includes the P wave, which is going to show us atrial depolarization, the PQ segment here. And then there's also some atrial activity happening during the QRS complex. You can't really see it though because the QRS complex is largely dedicated to the electrical activity of the ventricles. Ventricular activity includes the QRS complex, the ST segment. This is where that plateau is occurring in the last graph that I showed you, where calcium ions are flowing into the heart muscle, the cardiomyocytes, and then we have the T wave. All right, I'm going to cut it here and come back, and we'll start on the next slide. Thanks.